Hi everyone, welcome to another episode of my tutorials on Galaxy for Bioinformatics. This is the second episode in the series where I show how to run the DESeq2 on the Galaxy platform to analyze RNA Seq data and identify differential expressions. I will encourage you to check the first video. The link to that video is in the description box. By the end of this tutorial, you'll be able to generate these figures here. Okay, so we are going to generate all these cool figures here. And don't worry, I'll show you where to get the example data for this exercise. Now, let's get back to Galaxy. So what I want you to do is to register, okay? Create an account on a Galaxy platform and then log in. You can use this platform, use galaxy.org, or you can also use the EU one, that is use galaxy.eu. I'll leave the link to these platforms in the description box. I repeat, the links to these platforms can be found in the description box. All right. I will switch to the EU one. So what I want you to do is to create a new history. So come to this side, click on plus to create a new history, and then edit it to rename. So use the pencil icon here, click, and then edit. So I'll clear this name and then put mine there. So I'll say DESeq2 underscore analysis underscore 2008-2023. I want to keep track of my activities, so I will save it, and that is done. Now I want us to import the example data. So the data can be found on this GitHub repo of mine. There's the page, and I will leave the link to this page in the description box. So here, instead of clicking on each of the files to import, I have this text file which has the links to the files. All right, and don't worry, I'm going to give a brief description of the data later. But now let's click this link, and then you will have the download links for the files displayed for. So I want you to copy all of them. So highlight all of them. Okay, there are 7 in total. So copy, and then go back to your Galaxy platform, and then on the left side, come to upload data. So click it, and then when you come here, come to paste/fetch data. So click it, and then in this entry box, paste the links. And then when you come to the type, you see auto detect. Click, and then search for tabular. So type it, and you see it here. So there's a, so click it, and after that, come to start. So click on start, and then wait for the progress bar to turn 100%, and then check the right side. You will find some output here. So once you see that, close this, and then wait. Okay, at the moment it's in queue, so you have to wait for the background to turn green. All right, so we have to wait for the data to be imported for us. Okay, so now it's processing, so we have to wait. So let's wait for some time. Okay, the files have been imported, so let's take a look at them. File 1 to 6, these are counts, okay, read counts, okay, these are the count data. So each file represents a sample. Number 7 is the design tags, and it contains sample information. Now these files, the data set here, I obtained it from this study here. All right, I'll leave the link to this page in the description box. There's the paper. The data can be found on the Dataset database as well. So I'll leave the link to this page also in the description box. So if you take a look at the summary, the authors identified a tumor promoting long non-coding RNA. All right, they found that this non-coding RNA was regulating some pathways. All right, so for the experiment, what the authors did was to overexpress that particular long non-coding RNA, that is LETS1, overexpress that in some cells, and then the total RNA for these cells were isolated, and then that is what was used to generate data that we are going to analyze. I'm using a subset of the data. All right, so you can read more about this study here, and then just proceed. Okay, now let's get back to our Galaxy platform and then proceed. All right, now it's also important we examine the data, we do some explorations. I'll just open some of them here. let's just check them out. I think it's better we do that before we start the exercise. So I'll start with the count. So this the count, there's one of them. I'll just open this. So we have the count for the genes. I think I'll open with a spreadsheet software rather, so that we can see everything. Okay, so let's open that here. All right, so there's it. So we have the genes here and their respective count for this sample. The same applies to the other samples as well. All right. Okay, so that is it for the count. And then we also have design tags. Let me just open this. So the design tags contain information about the samples. Now the design tags was generated using this information, okay, the design table here. So the tags, let me just get, so the tags are used to identify the samples, okay. It lets us know the groups that these samples belong to. We will use this on Galaxy, all right. That is why I had to create this file. So if you also want to learn how to create files like this for RNA Seq Analysis, you can always reach out to me and then we can have a session on that. Now let's get back to the Galaxy platform and then proceed. So we are here now. So this is what we are going to do. We will need to link the data app. We need to link the design tags to their respective samples. Okay. So what we will have to do first is to create a data set collection. All right. So data set collections help us to treat files, groups of files as a single entity. All right. That's what we will do because we need to do the processing before we can run the DESeq2 on Galaxy. So when you come here, all right, so just come to this side where you see this checkbox, click it, and then select from file 1 up to file 6. And then come to the right side. I think I will zoom in a bit. Come to the right side where we have 6 of 7 selected. Click on this arrow here. You get a drop down. Scroll down and click on Build Dataset List. So click it, and then you have this coming up. And then come to Name. Click it, and then give a name for your collection. So I'll say Count. And then I'll come to Create Collection. So click it, and to be created. What you will see that the files will vanish. All of them are now here. Okay, so when you click Count, you'll find the files there. Okay, so that's what we have. Let's go back and then proceed. Okay, now what we need to do is to run the tag elements. So that will help us to finalize the linkage. It will help us to assign the identifiers to their respective data. And so you will search for tag elements. If you type tag, it will show up here, tag elements. You can also type tag elements in full, and it will also show up. All right, so any of them should be fine. So just type and then get it, and then click it. So when you click it, you will have this coming up. And so you will need to specify the input collection. Mine has been given here automatically. If you don't see yours, click and select it. And then scroll down to tag collection elements according to this file. So we will use the design tags file, which we have. So that's it. Okay, after that, come to Run To and then click it. So once you do that, check the right side, the data will be generated and output. So this is it. So click it, and when you click it, you will see this coming up with some colors. All right, so if you see this, then that means everything is okay. If you don't see this, please check and then follow the instructions that I have given. All right, now let's get back. Now we have finalized the data processing. All right, so now we are going to run DESeq2. So come back. Let's go to the left side. The tools again. Let's clear this and search for DESeq2. So this is it. Okay, DESeq2 determines differentially expressed features from count tables. So click it, and then DESeq2 will show up for you. So let's look at the parameters. The first one says select dataset per level. You will change it. So click and then select this one. Select group task corresponding to level. So click it, okay, and then you will have this coming up. We are doing it because we are using tags. We have done the process, and that's what we are using. We are using group tags. And then come to count file collection. So here we are going to use the output that we generated after we run the tag elements 2. All right, that is this one here. Mine is this one, and that is here. So make sure you also select yours. And then scroll down to factor. So click on insert factor. All right. Now, what is a factor? A factor, or let's say factors are the groups that can contribute to the differences in gene expression. All right. Now, the study that I showed, let's just use the design table. I think that will be better. So the study that I showed, they had cells that had overexpressed LETS1, and they also set up control groups. All right. Let's just use this one here rather. Yeah, so we have the control groups, and you also have the cells that have the overexpressed LETS1. So due to the difference in treatment, this can lead to a difference in the gene expression patterns. Okay, so it can lead to differences in gene expression. Okay, so in this situation, group becomes a factor. All right, in some experiments, you will see treatment or some other stuff, but that's what it happens. So this is a factor. Okay, that's the group name, and then these are the subgroups. So we have control and TGF beta. These are factor levels. So we have factor and we have factor levels. All right, let's go back to Galaxy. So let's go back to Galaxy, and then let's specify a factor. So here the factor is what? Group. That's factor name. That's what we use, group. And then factor levels, we have control and TGF beta. All right. Now, when specifying the factor levels, the control should be the last one to be specified. All right. So we are going to click on insert factor level, but we will select the TGF beta first. Okay. The control is a reference, so that becomes the last. So we will click on insert factor level, and then you type TGF beta. Okay, so I'll click and I'll say T. Let me just clear it, do it here. So I'll say TGF beta. Okay, and I'll click on this. Okay, when I click on this, you have the tags shown. So the tag I want to use is TGF beta. So I'll unselect or deselect the control one. I want this one. So I'll select it to get it. So that is it. Now I'll come to insert factor level again. And this time this will be control. So I'll come here, select all, select all. You can even click here as well. Okay. And then you can select control. Right. So just make sure to select the tags, and then you are done. All right. So this one is a single factor. You can also have analysis which involves multifactors, where you have 2 or more factors. I've covered one such tutorial, and so you can watch that tutorial. That's the first tutorial I made on DESeq2 on Galaxy. All right. So check that tutorial out if you are looking for multifactor analysis. All right. Now we are done. So what we have to do is to scroll down. Let's check some stuffs. Yeah, the choice of input data, make sure it's count data, e.g. from ATC count, feature count, or string type. So that's what we are using. All right. And by the way, for counts, I have a tutorial on count. So if you're looking for a tutorial that can help you to generate your read count or gen count, then check the description box. I have a tutorial on that. So you can watch that to get started. All right. Now let's run DESeq2. So click on run 2. And when you click it, you should see some output generated. At the moment, the job is in queue, so we have to wait for everything to get done. So once the background turns green like this, that means that the outputs have been generated. So that's what we have to wait. So let's wait for everything to get done for us. Okay, DESeq2 has completed its work, so we have the output here. All right. You can also generate additional outputs, but this is to help you get started and you can explore the other options available. All right. So we have this result file that you can click. Okay. So you have this one here. It has the genes, and you also have the base mean, log2 fold change, and you also have additional information. All right. So a description of these headers can be found on the DESeq2 tutorial page. All right. And so you can download it. Let's just download this one. I will just download here. So I will download it, and then I'm going to explore it. It's currently downloading, so I'll just leave it for now, and then I'll check it out. Okay, the file has been downloaded, so I can check it out. Let's just check it out here. So there's it. All right. So this file can also be opened with a spreadsheet software. I think I'll do that, and then we'll see. So let's open it. This is it. So we have everything we need. Okay. It's also sorted for us automatically. Now, the headers are missing by default. So before you download, you can just add the headers. Okay. So if you add the headers, you'll get something like this one here. Okay. This is an example. Okay. This one, I generated it when I was preparing another tutorial. So you can get something like this, and then you can do your downstream analysis. All right, so that is it. Now let's get back to Galaxy. In addition to this, there's also another file. Let's just close this. We also have this one. This is a PDF that has some plots. Okay, so this plot will help you get started in terms of the analysis. So you can save it. You can see that it's being downloaded as well. Okay, so we can wait for it to complete, and we can also explore that. Okay, so the file has been downloaded. So we have the output here. So we have a PCA plot. Okay, now this PCA plot is weird. Okay, I tried it on my machine as well, my local computer, and I got something similar. So when I checked online, the suggestion was to manually extract the PCA values and then plot with ggplot. That one worked for me. So you can also try that. All right. So I think I'll also try to contact the Galaxy team to see if there's something they can do when you have some situations like this. So for the most part, the PCA plots always look good, but we can have situations like this. So if it happens, there are workarounds. All right. So you don't have to worry much about this. You also have sample to sample distances that can help you to compare the gene expressions for the samples. And we also have the expression estimates. We also have histogram for p-values for the groups. We also have MA plots as well. So these are the plots that Galaxy generates. In addition to this plot, you can also generate volcano plots. You can also generate box and whisker plots. You can also generate MD plots. You can also generate UMA plots. You can also generate heat maps of log, transform, normalized counts. And these plots, I have scripts that can be used to generate that. So if you want those scripts, you can check the description box for those links, right, to the scripts. All right. Now, after identify the differentially expressed genes, and so all these things will help you to visualize the data, and then that will help you to explain the results to others. It will also help you to understand your data. All right. Now, aside from these plots, all right, after identifying the differentially expressed genes, what you can also do is to perform functional gene arrangement analysis, and you can also perform Kirkpatrick analysis. So these activities, I'm currently working on tutorials for them. So I will encourage you to subscribe to this channel and get a notification when those tutorials are uploaded. Now, if you also want to learn how to run DESeq2 on your own PC using R, then I will encourage you to check the video right here.